everyone. So it's 8:45, so I think it's time to slowly get started with the first session of today. So also good morning to the online audience. So it's great to have you with us today as well. So this morning we're going to have a session on image analysis. So we've heard for the past few days about amazing microscopy techniques and technologies, and we've seen that people are now acquiring ever more complex data sets. And we've also seen that one of the bottlenecks is often now image analysis. And it's really the aim of this morning's sessions is to show some of the state-of-the-art advances that are now available to everyone to analyze your data. And on this, so it's really my great pleasure to introduce Anna Kreshuk. So she's working at EMBL as a group leader in the cell biology and biophysics unit. And her research really focuses on using machine learning-based methods to the analysis of biological images. So you probably know many of the tools that a lab and a research has been creating over the years, and in particular Ilastik, which is a fantastic tool for image segmentation. Today, Anna will be telling us about image segmentation with little training data, which is, of course, very important for us biologists. So, Anna, thank you so much for joining us here in Turku, and then please take it away.
Yeah. Thank you very much for the introduction and for inviting me to come here. It's a real pleasure to be here in person. So this is not exactly what I wanted to show. Yeah, that's, yes. So let's talk about image segmentation. And the work I'm going to show today has actually been done by all those great people over there who are all very interested in image segmentation, but also in other questions. But today I want to talk mostly about where we are going with our segmentation algorithms. Right, and so since I started the lab, we've been doing a lot of different segmentation tasks, right? So we've been doing things for electron microscopy. We are still doing a lot for electron microscopy. We've been doing things for light microscopy, especially for membrane-labeled cells. And we've been doing more things in light microscopy. So this has all been really fun, right? And I feel like we have developed a lot of interesting tools in there. And yeah, of course, there's also Ilastik that Guillaume has already pointed out, right? Which is trying to bring this kind of machine learning-based tools to people who don't have machine learning expertise themselves, right? But you know, so there is a certain delay between the things that we develop as research and things that do end up in Ilastik then completely usable by the non-computational biologists. And what we have noticed, like the state to which we have come with our best segmentation algorithms, is that we can make it work very well, at least given enough dense training data. Right? So if you have a sufficient amount of ground truth for a segmentation problem, right, and this is a very big if, we can get it quite far nowadays. But the problem is that, of course, we don't want it to only work on our hands. And we also really want to go away from the requirement of dense training data because, well, if you have ever done segmentation in 3D and had to provide ground truth for it, you know that this is a process that really takes months of annotation and tens of thousands of euros. So this is something we don't like, and we also really hate it when we have to go back to collaborators and ask, like, we need just a little bit more, and you see this horror in their eyes. So we try to actually reduce this requirement, and that's partially what I'm going to tell you about today. Right, and the first project I wanted to show is what we call SPOCO, right, which stands for Sparse Object Level Supervision. And yeah, so it's a long title, but what it actually amounts to is that right now we are training with data that looks like this. Right, so what we actually want to train with is data that looks like that. Right, so we want to be able to train from images where you have just a few of the objects annotated and don't have labels for the background. Right, so this is the so-called positive unlabeled setting. And in this, so it's the paper is long and technical. Without going too much into technical detail, let me just say that we have found a way of how one can stabilize the training from very few examples by going to the big unsupervised parts of the data and introducing consistency constraints. Right, and in there, you can see that in this small but difficult example, so there is this benchmark data set of segmenting plant leaves, it already does pretty well with just 10% of the training data. Right, so there is still one error left in there. Can you see my cursor? Yes. Yeah, so there's still one error left in there, but it doesn't take much to correct, and we only use 10% of the training data. And of course, it's, we actually want to use it for microscopy, not for plant leaves. And you can see that also for EM, it works quite well, right? So if you take in a transfer learning setting, which for many things is actually the most interesting one, right? So if you already have a pre-trained network, and now you try to use it on your own data, and you get results which look like this, right? Which is not what you expect mitochondria to look like. And then you say, okay, let's just use 5% of the data on the target domain, and it gets so much better, right? And we can do another example like this, and you see it even starts to recognize this really tiny objects that we didn't have in the original training set. So the same approach that we call SPOCO, right? So it works for EM, it also works for light microscopy, where we can also transfer between data sets which to a human look very similar, right? But to a machine would actually look too different, right? Because of all the intensity differences between the two microscopes and two independent labs. Yeah, so we can now train on one and then just transfer to the other with a little bit of new training data. Yeah, so there's still a lot of open questions in this. One is like actually making it scale to really big data sets, because yeah, light microscopy, especially light shield microscopy, is now getting very greedy in this sense. Yeah, we have to optimize the clustering step to be faster and also less memory hungry. And, but I think the biggest challenge that remains in there is the integration with annotation and analysis tools. Because in Ilastik, we don't have an easy way to label complete objects, but we are thinking about it. We are talking to people. We are like looking into Napari that has ways of how you can sparsely label objects. So this would be, so I think this is an approach that works really well, and also in terms of theory is quite elegant. And the next thing that I wanted to show to you is something that is much more based on the Ilastik model of annotating things, so just giving brush strokes, is not as theoretically elegant, but I think is actually very usable in practice. Right? And yeah, so this is the one that we kind of tentatively call from shallow to deep, but unfortunately this name has been used so much now that yeah, you can't just Google for it and find it like this, but I'll show you where to find it. Yeah, so if you recall how shallow classifiers work, right? If you think back about to Ilastik, right, where you give brushstroke labels, it's very fast, it's very interactive, right? It can digest your labels almost immediately. And this is something that we have been really missing on the deep learning side of things, where you have to provide this very big, densely labeled data sets to get to the good results, right? And unfortunately, of course, there are also downsides to this, which is that random forests are not as powerful, right? And all of these shallow classifiers are not as powerful. So if you take an example, again, of mitochondrial segmentation in EM, but you can substitute this for your favorite light microscopy question, right? You can see that, well, if you train a random forest, and if you train carefully, the results will be a bit better, but still, it's not exactly what you would want to have, but you can get them so fast, right? So we've spent quite some time thinking of, well, how can we actually reuse this probability map that the random forest produces? Because it's clearly taking you somewhere. There is a lot of information encoded in this, right? So how can we get this data that the users can generate so fast and actually digest it into a better classifier that would, take it another step up, but without requiring weeks of annotation and another weeks of training time? Yeah, so we tried the most straightforward way, which is to just use the random forest predictions as pseudo labels and hope that even though these predictions are very noisy, that we just can have so many of them that it will not really matter. And yeah, that was not good enough. Right? It gets a bit better, but it's not really, there is still too far to go, let's put it this way. We have tried to use the pre-trained CNN features. This is an approach that was also quite popular before 2020 or so. So where you would pre-train a CNN on some very big data set, especially in natural images, and then take whatever it is they learned and then put that as features into your shallow classifier. Because then the user still only has to label a little bit on the images, but you can kind of use the power of deep learning more. So we tried that. This is also, in our opinion, not good enough. Right, so it does get better than completely generic features that we put in there, but this kind of difference is not the deal breaker that we would need to justify the investment of time into putting the CNN into this. Right, and so what we actually came to, what I think works pretty well, is the realization that you can't use these predictions as labels. Right? You also can't really use them as features, but you can use them as an intermediate step in training. And the deep observation that led to it all was that the data sets actually look very different, or too different for the neural network. But the probability maps that the shallow classifier or whatever other segmentation method you use produce look very similar because you are looking for the same thing. They will be differently wrong, but there's still a lot less variability in them than you would have in microscopy itself. For example, there are no intensity variabilities. And this is, what is the whole idea is actually implemented in this paper that I'm showing there, exploiting feature-based classifiers for domain adaptation and semantic segmentation. So what the domain adaptation there means is that you are not really starting from scratch, right? But that you have something that was trained on a similar task before, and now you want to adapt to your own data. Yeah, and so the way that works is if you recall the left part of the figure, right? So you have your favorite shallow classifier. Well, for us, of course, Ilastik. For you, it could be, I don't know, LabKit, Veka, whatever. And you train it, and you have a probability map that, like we said, is maybe not so good. Right? So what we do on top of this is we take a neural network, and we train it to improve the random forest predictions. And the important bit there is that the neural network never actually sees the raw data. Right? So what it sees is just these two parts. Right? It sees the crappy random forest predictions, and it sees the ground truth. Right? And then it knows how to turn this into that. But it doesn't know what raw data was there underneath. So it's actually agnostic of all these slight differences in the noise distribution that you have between different microscopes. And this enhancer, because it doesn't see the raw data, is actually sufficiently robust to be used on different data sets. So what we hope for in this is to take the sufficiently popular tasks that have a volume of ground truth and train on those, such as, for example, this mitochondrial segmentation problem has the MITO-EM data sets and also some new data sets that Kedar has recently made available. So we would then train this enhancer, and you could then apply it without retraining. Right? So because at test time, the way this would look is that you take a new data set, you apply your labels Ilastik style, right? Then you get your new predictions, which are, well, yes, still a bit noisy, but now looking sufficiently close to what we had before. Right? And then you apply this pre-trained enhancer network without doing anything, right? It's just one inference run. It's super fast. It can be interactive time. And you get a much better prediction. So this feels like it's almost coming for free, but you can actually make it even better. Right? So what the next step you can do is that notice that the enhancer predictions are not so bad, and they can be used directly as pseudo-labels. I mean, they're still a bit noisier than real ground truth, of course, but they are sufficiently good to train a network now as if they were ground truth, because we have so much unlabeled data, and the enhancer predictions we can produce for anything, and that means that we can have a lot of this data now, and we can train an even better network. And this is the one that we call segmenter. So we tried it on different tasks, right? So it actually works very well for this mitochondria, right? And especially the final segmenter network really makes it much better. So you can see in this example that, the random forest, well, we gave few labels. I didn't do so well, so the enhancer has really made it better. But then when you put another effort in running it with these labels with these predictions as labels, it gets even better. And this part, actually, going from the enhancer to segmenter, right, the part that I just showed here, this doesn't require any, let's say, intelligence in running it, because we already know where all these hyperparameters have to be set. Right? It's just a question of submitting a job, and then it comes back. Right? But you don't have to really tune anything. You don't have to deeply understand what is happening. You don't have to manually switch things around. Yeah, so this is where it is now. You can see, so this is a measure where higher is better, right? And you can see the very big jump it makes from the random forest, and for very different data sets, because mitochondrial segmentation is a popular problem, so we could really test it around on very different approaches. Yeah, it also, if you look at the boundaries, this is also for EM, and the reason why we always go for EM in this method development is that EM is inherently a more difficult problem because the background is so structured. But of course, it also works for light microscopy. And here you can also see that the random forest tries and is not so bad, but of course, if you put this enhancer on top, it gets much better. And then if you train for another iteration, it gets even better again. Right, so one thing that we kind of didn't plan for in the beginning, but this works, and I find that this is kind of cute, is that because the enhancer does not see the raw data, you can actually do cross-model training, as long as your objects occupy the same amount of pixel space. Right? So, like, for example, here you have the boundaries in EM, and here you have the boundaries in light microscopy, but in terms of pixel size, they are about the same, right? Like, I don't know, 2 pixels across, 3 pixels across. Right? And that means that you could actually take an enhancer that was trained on this data and apply it to light microscopy prediction, and it still works, right? It still enhances and does it better. Of course, you have to remember that these kind of things have to be used responsibly because it doesn't see the raw data. It's mostly going by shape. And it's used to enhancing a very noisy prediction, which means that if you actually try to do not cross-model, but cross-task application, what you will have is like, suppose you have this data set, right, and you trained an enhancer to do mitochondria. And then you, instead of training the random forest to do mitochondria 2, you train it to do boundaries because maybe you forgot what your enhancer was doing. It will find you mitochondria again. Right? It will find the little pieces of mitochondria that are still present in the boundary prediction, and it will enhance them all the way. And they are even at the right places, most of them. But this is, I guess, not what you would mostly want to do. Of course, it also goes the other way, right? If you start with the boundaries and then try to enhance the mitochondria signal, it will find you the boundaries. And some of them will even make sense. Like, not all of them, it will not be a perfect boundary prediction, but it will find the little there is of the boundary signal in your predictions and amplify them. So be careful. And make sure that when you're using an enhancer like this, you're actually enhancing for the same task. Yeah, so this approach is so well compatible with the Ilastik annotation model that this is something that we hope to actually bring out to you soon. We hoped it to the organizers of the Swiss Image Analysis School ZIDAS that we will have it there, and this is early July, so really soon. And I mean, this is already a prototype, you can see. And yeah, so the way we imagine this will work is that you will just have this one little additional menu here for getting a pre-trained network in. And then you give your labels like you usually would in Ilastik. And so this is very few labels. Normally you would give a bit more and get a better prediction. But if you don't even want to bother with this, you give very few labels and you get a very nasty prediction. And then you just apply the enhancer and it all gets better. So that's how we hope it can work for users that don't want to train the networks themselves, but can enhance the random forest predictions with the power of deep learning, so to speak. Yeah, so then I hope the question that you're all asking yourselves is now, well, where do I find all these pre-trained networks? Right? Because if you want to do anything that is transfer learning like, you will have to have a pre-trained network that you are starting from. Right? And usually the first training of the network is the one that takes all the skill and all the training data and all the computational time. And fine-tuning it to something else doesn't take so long, and like we have shown, you can do it with very little training. So where can you find all the pre-trained networks? And this is the project that I'm very happy to tell you about because it was actually born at ELMI in Brno. That was, what, 3 years ago now? And this is also due to ELMI being such an open conference on the one hand, and at the same time allowing for a lot of discussion time. So I met Wei Yang there, and we, in this whole free afternoon, converged that we actually have the same idea of what we need to get as a community, that we need like a place where people can have their models all together, and we can share the pre-trained networks. And what is especially important is that we can share the pre-trained networks in a way that we can use them in each other's tools. Right, because we have a lot of people who are developing methods and a lot of people who are developing user-facing tools, and they all have their own little garden of pre-trained networks, and not being able to exchange them is very limiting, both for us and for the users. And this is how starting from Wei and myself, the whole community came together, and you can see all the partner logos here, probably even not all. And we all came together with a vision to have a place where we can share everything that is related to having neural networks for microscopy data, so both the pre-trained models, but also their training data, and also the code that trains them. And here you can see the community as it stands now. We are happy if more people want to join, but you see, it's a lot of people, it's a lot of popular tools, and yeah, so many of them here today. And yeah, so we hope that with this, we can really have a common repository where this kind of pre-trained nets that we would then train on the data of our collaborators, and you would train on the data of your collaborators or on your own data, can then make its way for everyone to use. And well, when I say for everyone to use, this is not only in the sense of like it's publicly available for download, but really that the networks are stored in the same format so that you could download the same thing and use it in both DeepImageJ and Ilastik, for example. Right, so what is in terms of like what is currently there are already a lot of models. Let me try to maybe just show you the website quickly. Right, so you see this is what it looks like. Right, the address is bioimage.io, as you can see here. And yeah, so we have some documentation already, and we have the community tools and their models, their applications, which, well, usually means notebooks. We have data sets, right? So here are all these kind of different models, and you can see that we here have you can see the examples here, and you have what we call the model cards, right? And they all have a DOI, and they have a memorable name, like the late big lobster, right? And you can see who created it, and there is a lot more information in here, and you know how to cite it, you know what it was trained on, you know how it was trained, and you can even do things like try it out yourself, right? So let's see if that would really work. So worked for me in the morning. Let's see if it's still running. But that is very internet dependent. So let's see how fast or slow this would be. So this is the test run functionality that we have implemented for people to kind of quickly get an idea of what the network is doing and if it would fit their data. And you can put in your own data, or you can take our test input files that we put in there so that we know that all the models actually work. Right, and we can select this file now, and which one did I have? 3. Okay, and then we can say that, yes, that's the one we want. And then we submit this, and we see what this network will now predict. Right? So that will be the prediction. Here you can see that it fits and that the network is actually functional. So if you download it for use in Ilastik or in DeepImageJ or in any other tool of your choice that we support, right, you can do the same thing and now on your own data. You can also upload your own data here, but it should not be too big. Okay, so you have seen we have the very big set, well, not very big, but let's say we have some networks, quite a few. And the most important bit of it is that it's all done in a format that is understood by very different tools. Right, you can test run the model like I just showed. It works for most of them. The few where it doesn't work are the ones that were done with the very old versions of the deep learning frameworks. We will fix those too. It's just, takes a while. You can see how things were trained. So if you wanted to actually go on the path of training things yourself, there are some very good pointers in this, because for most of these models, you can also have the notebooks that were trained in them. There are also zero-cost notebooks that show examples of how you can export a model into this format so that you can immediately upload it for everyone to enjoy. And so this was kind of a hobby project for all of us there for a couple of years now. But we have now also received funding from the EC to keep going with this. And this is a project ambitiously called AI for Life. And there we want to make the Model Zoo like the real service for users and developers, building on what we have now, but also expanding it a lot to make it more convenient, both to submit models, but also to take models. And there are a lot of technical challenges in this, right? In actually running the things, but also in the things that we have all discussed as a community for a long time, such as how do you submit annotated data? How do you choose the problems for which you want to annotate the data to make the largest impact in the community? So these are all the things that we would actually really like to do together with you, right? We will have open calls for this too. And the whole project is actually coordinated by EuroBioimaging, so I'll be at their booth and we can chat about this in the coffee break. To summarize, I think segmentation is getting easier, and overall the field is making a lot of progress and making it better. So it's so we do a little part in this, but there are also many other people in the community that have really worked on making this a solved problem. Right? We have seen that shallow and deep can be combined, and it can be very advantageous in cases where you don't want to spend too much time annotating your data. Pre-trained models are very valuable because of this, and you can find them at the BioImage.io. So come and look at them and share yours. Thank you very much.
Thank you, Anna, for a wonderful presentation. And we also have time for questions. So any question from the audience? So over there from Ricardo.
Hi, Anna, great talk. My question is regarding the enhancer network. I guess from a user perspective, the user never knows how much annotation they need to give in Ilastik for the random forest to generate a statistical map that is good enough for the enhancer to go on. Are there ways to actually know how quality control scales with annotation? Is that something that the user has some perspective on?
Well, nothing that would be very user-friendly to interpret. Right? So you can look at the distribution of the two maps. Right? So you can see if the probability map that you're generating now is close enough to what the training was done on. But the problem is that will be some measure of distribution similarity, which is not very easy to interpret. And also, in the end, what matters is not the distribution of the data as it goes in, but the distribution of the thing that actually passed through the network. And this is hard to estimate, which is why we think that just like it is for the random forest in a way, right? So how do you know when enough labels are enough? And you know because it stops improving, right? Or because it's already good enough. And this is how we also hope to do it there. We will rely on the humans interactively looking at this, because you will be able to because the enhancer doesn't need to be trained, right? So it can be applied real time, and then you can train the random forest classifier, apply the enhancer, see if it's good enough. If not, you add more labels, or you add more labels in the places where it's still wrong. And in Ilastik, this has worked really well for many years now, so we hope we can just remain with the same idea, and then eventually, once the science progresses to give easier quantitative indicators of things being good enough, of course, we will incorporate them. Thanks.
All right, any other question? One over there. It was at the back first, and then.
Yeah. Okay, sorry, I stole the microphone and I jumped in front. Just quick question, because super interesting work. What I was wondering is that, in a way, your initial step is sort of a normalization. You're changing from one space to the other. Does the user have to have any knowledge as to which features were selected in the shallow one, or do you think that that's not particularly important?
No, I think that's not important. What is important is that the predictions are, let's say, sufficiently similar. So, for example, what would not work is if you have one classifier that was done with like 5 classes, right? So that you have like one distribution in the probability map, and then you try to apply it to a classifier that only has 2 classes, because that will be a little bit different. Right? What features were selected, I think, is less important, because it doesn't influence the appearance of the map so much. Also, the number of random forests, like all these things, I mean, that we have actually tried, and this doesn't matter.
Okay, one last question from the back.
Hello, thank you for the very interesting topic and very nice presentation. Question about this platform that you showcased, and you mentioned that you are improving the algorithms. Do you happen to have or thought about some kind of a version control of these algorithms, or is it already implemented? I'm thinking about, for example, when you are doing manuscript, that you might want to report that if there's a changes in the future and you want to go back, like, of course, in 3 years, you can always go back to the certain version that was used back in the days when you made it.
Thank you for this great question, because this is the thing that I should have talked about but didn't. And yes, so this kind of reproducibility is definitely one of the reasons why we have started the whole thing. Because you often read about, and then we apply a neural network. And then what, how, what it was. So here you have the full traceability, because it has a DOI, and the DOI is actually attached to the set of the weights. Right? And then if you update the weights, for example, if you get more training data, or if you just train for longer, or if you get a clever idea of how to change the learning rate, you will get a different set of weights that will have a different version then, that will have a different identifier, and you can link to the previous one so that you know how they are all connected in time. And we really hope that with all of this, things will get a lot more reproducible. And you also will, because we have the test environment, you will also know that the previous one runs. And you'll be able to immediately then compare the different versions, and to compare multiple models trained on the same data, and at the same time to compare sort of architecturally the same model but trained on different data sets or on bigger collections of data sets. So yes, making it all fair in the political sense is a big part of starting the whole enterprise.
All right, thank you. So any question from the online chat? No? Okay, then join me in thanking Anna again for a fantastic talk, and then we can continue the discussion also in the coffee break.